Welcome to our educational channel. In this video, we will learn about properties, nomenclature, and classification of enzymes. Enzymes are specialized proteins that can act as biological catalysts. Catalyst is substance that accelerates the rate of chemical reaction without itself undergoing any change in the overall process. The substrate is the substance on which an enzyme acts, and it converts into the product or products. Now let's see the properties of enzymes. Almost all enzymes are proteins except ribozyme, which is RNA molecule that acts as catalyst. They are colloidal in nature, heat labile, and water soluble. They are specific in their action. They can be precipitated by protein precipitating reagents. They contain 16% weight as nitrogen. Nomenclature of enzymes. The International Union of Biochemistry and Molecular Biology, or IUBMB, assigns each enzyme systematic name and an enzyme commission number. A systematic name is a way of naming an enzyme based on the specific chemical reaction it catalyzes. For example, the systematic name of alcohol dehydrogenase is alcohol NAD plus oxidoreductase. This indicates it oxidizes alcohol using NAD plus as the electron acceptor. Enzyme commission number. It's a 4-digit numerical code that provides a universal hierarchical way to identify enzymes, ensuring clarity and consistency in scientific communication. The enzyme commission number consists of 4 digits, each representing level of classification. Take, for example, an enzyme alcohol dehydrogenase with enzyme commission number 1, 1, 1, and 1. The first digit represents class of the enzyme. The second digit represents subclass of the enzyme. The third digit represents subclass or subgroup of the enzyme. The fourth digit represents unique or serial or specific number for this enzyme. The IUBMB classifies enzymes into 7 major classes, each defined by the type of reaction they catalyze. These classes are assigned number from 1 to 7, forming the first digit of the enzyme's enzyme commission number. Let's discuss each class separately. Oxidoreductases. These are the enzymes that catalyze oxidative reduction reactions. And there's transfer of hydrogen and electrons or removal of oxygen. For example, alcohol and NAD plus in presence of alcohol dehydrogenase gives aldehyde and NADPH plus H plus. In this reaction, the hydroxyl group of the alcohol is oxidized. Specifically, a hydride ion is transferred from the carbon atom bearing the OH group to NAD plus and reduced to NADH. Hence, there is both oxidation and reduction in this reaction. So, the enzymes that catalyzing these reactions are oxidoreductases. Examples of oxidoreductases are dehydrogenases, oxidases, oxygenases, peroxidases, and reductases. The transferases are class 2 enzymes. They catalyze transfer of functional groups except hydrogen to acceptors. In the given reaction, there is transfer of phosphate group, which is a functional group from ATP to hexose, converting to hexo-6-phosphate in ADP in presence of hexokinase or glucokinase. These are some examples of transferases. Hydrolases are class 3 enzymes. These enzymes catalyze hydrolysis of various bonds by adding water, as shown in the given example. The main hydrolases are digestive enzymes like amylase, maltase, pepsin, trypsin, and lipases. Lyases are class 4 enzymes. These enzymes can remove groups from substrates or break chemical bonds by mechanisms other than hydrolysis or oxidation. In the given reaction, fructose 1,6-bisphosphate is cleaved to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate by aldolase enzyme. These are some examples of lyases. Isomerases are class 5 enzymes. They catalyze the rearrangement of atoms within a molecule, converting it into one of its isomers without adding or removing any atoms. In the given reaction, glucose-6-phosphate is converted to fructose-6-phosphate by phosphohexose isomerase enzyme by isomerization. These are some examples of isomerases. Ligases are class 6 enzymes. These enzymes link two substrates together, usually with hydrolysis of ATP. In the given reaction, pyruvate with HCO3- is converted to oxaloacetate by pyruvate carboxylase enzyme by hydrolysis of ATP. These are some examples of ligases. Translocases are class 7 enzymes and latest to be added in the list by IUBMB. Translocases facilitate substrate-like ions, sugars, and proteins movement across membranes or within cells, utilizing energy from ATP or GTP hydrolysis, proton gradients, or redox processes. Examples of translocases are ATP-binding cassette transporters, calcium channels, and sodium-potassium pump. If you found this video helpful, please like, share, and subscribe.